Now we are going to discuss about restriction endonuclease. RE. We will denote generally RE is restriction endonuclease. So the term restriction endonuclease, if you split the term endonuclease into two words, endonucleases. So you know that the nuclease is an enzyme which can cut the nucleic acids. Okay, nuclease is the enzyme which can cut the nucleic acids. Plus endonuclease means, say suppose this is a nucleic acid, that is DNA. DNA means deoxyribonucleic acid. So this is 5 prime, this is 3 prime. If the restriction endonuclease, it means if your nucleus cut the DNA in the middle, not at the ends of a DNA, then that type of nuclease is called as endonuclease. Contradictory this one, exonuclease. So here, exonuclease is the enzyme which can cut the DNA at terminal parts. Okay, so endonuclease the enzyme which can cut the nucleic acid at the intermediate parts are in the middle. Okay, so here endonuclease means the nucleases, the enzymes which can cut the DNA in the middle portion or intermediate are here, not at the terminals. Okay. So restriction, what are restriction endonuclease? What is the significance of restriction endonuclease? Okay. So coming to significance and usage and application, these restriction endonuclease will use in RNA technology or gene manipulation and different fields of biotechnology for cutting the DNA. That's why these are also called as molecular scissors. That's why it's also called as molecular scissors. Okay. So how they will cut and what is the basis for restriction endonuclease? How this property came to restriction endonuclease we see now. So first, generally these restriction endonuclease are present in prokaryotes. Especially till now, whatever the restriction endonuclease retrieved or separated or extracted from bacteria. Okay. Say suppose if you take an example, restriction endonuclease extracted from E. coli, Escherichia coli is EcoR1. EcoR1 is the restriction endonuclease extracted from E. coli. Okay. So suppose what is the function of restriction endonuclease in the bacteria? Say suppose if you take a bacteria E. coli, it's a classmate. Okay. Any foreign particle, foreign DNA like bacteriophage attacks bacteria. Bacteriophage is the viruses which can live on bacteria. And whatever foreign DNA comes from outside can be cleaved by this restriction endonucleases. The function of restriction endonuclease is to cleave the foreign DNA. And these will have another property called methylation. They will not recognize the self. So suppose this is the nuclear, this is the genomic DNA, this is the plasmid DNA of bacteria, and this restriction endonuclease no longer identify the genomic DNA. Why? Because methylation takes place. Okay, that is called a post-restriction modification system. Okay, so this is the bacteria is the host, and it will restrict the entry of new DNA and it will modify the own DNA by methylation so that the restriction can be done for the foreign DNA, not for the own DNA, or cleavage is for foreign DNA, not for the own DNA. So we can say that these restriction endonucleases are specifically cuts the DNA. Specifically cuts the DNA. Okay, then how we will use this property in our RNA technology we will see now. So here, eco-argon restriction endonuclease, it has a sequence called recognition sequence. That is GAATTC. Okay, if you say the complementary sequence CTTAAG. If you did it, it's a 5 prime, it's a 3 prime, it's a 3 prime, it's a 5 prime. So the restriction endonuclease of extracted from eco-argon can recognize the sequence GAATTC in a DNA. So suppose this is a DNA. Whenever it recognizes the sequence called GAATTC, it will bind to that and it will cleave that. So coming to cleavage part, this is cleavage site. Okay, it will recognize 6 bases, 1, 2, 3, 4, 5, 6 base recognition site, and the cleavage site present within the restriction site. Okay, so now once the cleavage occurs, if you see the product is GAATTC and CTTAAG. So cleavage occurs here. Then the product is DNA will be cleaved like this and it will produce overhangs. Okay, so what is the use of presence of overhangs and all we will see in the next video. So coming to nomenclature of restriction endonuclease. Nomenclature. Okay, so if you take the say the eco-argon extracted from eco-right and how they are given the name eco-argon, the first name of genus name will be the first letter. First name of the genus. So here Escherichia coli. E. coli means Escherichia coli. This is genus name, this is species name. And first letter of genus name will be the first letter of the restriction endonuclease. And the second, first 2 letters of species name will be the second subsequent letters of species name. And R is the zero type or subtype. 1 is the sequential number. When say suppose there are 2 or 3 restriction endonucleases extracted from the same strain, eco-R1, eco-R2, eco-R3, eco-R4. So R is the strain. CO represents the species name, first 2 letters of the species name, and E represents the first letter of genus name. Usually first letter will be the capital letter and second 2 letters are small letters, and serotypes will be denoted in the capital letter again, and sequential numbers in the Roman numbers. Okay, this is how the nomenclature of restriction endonucleases. So there are 3 types of restriction endonucleases are there. There are type 1 restriction endonuclease, type 2 restriction endonuclease, type 3 restriction endonuclease majorly, and type 2S, type 4 type of restriction endonuclease also identified. So these differences, this classification based on the site of recognition and also mode of action and the morphology of the agent. So the types of restriction endonuclease